Okay, folks, I'm here with my puppies and my kitty, and we are all here, and we're going to do a little model of what is happening in cell division in eukaryotes, namely mitosis and meiosis, because this is a thing that can be very confusing. So eukaryotes, one of the things that's distinctive about them is that they have these specialized types of cell division called mitosis and meiosis, and one of the things that happens there is we've got these linear chromosomes that are separated into discrete units. And a chromosome is any individual molecule of DNA. And that leads to some confusion because sometimes we can have a pair of sister chromatids, which are exactly identical copies of the same chromosome, and we might say that that's a chromosome. The pair of sister chromatids represents one chromosome. But when they separate from each other, they each become their own chromosome. We can also say that homologous chromosomes are the same chromosome in a sense. So I'm going to try and tease all that apart as I'm doing this little card trick for you. It's not a magic trick, it's a biology trick. So here we go. So in this case, I'm going to use these cards to represent a diploid cell, okay? And right now, this diploid cell is in G1. It's in its growth phase. It has no copy DNA. All the DNA is condensed into chromatin, which it's hard to model using these playing cards. But if I tease it out, you'll see that I've got ACE2-3 of clubs, ACE2-3, and ACE2-3 of diamonds. And the suits, clubs and diamonds, are going to represent the parentage of this cell. So these can represent dad's chromosomes, and these are going to represent mom's chromosomes, okay? And the ranks, so the two aces are going to represent one homologous chromosome, the twos are going to represent a second homologous chromosome, and the threes are going to represent a third homologous chromosome. For this particular cell here, we would say that N, the haploid number, is going to be 3. But this cell, because it's a typical animal cell, we'll say, is already diploid because it's got 2 copies of each chromosome, ACE2 and 3. Okay, so we can't go through mitosis just yet because, as I said, we're in the G1 phase, which means we need to replicate our DNA, which means I'm going to have to do a little bit of adding some extra cards in here. Is that enough? Well, let's see. We'll throw them all in there and scrumble them up. So that's DNA replication. So we've just gone through S phase of interphase, and we're going to go through growth 2 phase, and we're going to enter into mitosis. So what's going to happen in mitosis is the chromosomes are going to start to condense, and I'm going to try and line them up like we have for metaphase. So you can see we've got 2 copies of the ace of diamonds now. We should have 2 copies of the 2 of diamonds, 2 copies of all of these cards. So I'm going to try and put them all in here so they're all lined up. And it doesn't really matter so much the order that I'm putting them in, as long as every single card has an exact copy. So now each of these represents a set of sister chromatids. 2 copies of the ace of diamonds, 2 of diamonds, ace of clubs, 3 of clubs, 2 of clubs, 3 of clubs. Okay, and if it makes you feel better, which it might, I'll go ahead and put them all into an order that makes sense, okay? So what's going to happen at anaphase of mitosis is the sister chromatids are going to separate from each other. Bloop, 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 bloop. And we'll just put these all together into one daughter cell, and we're going to say these are the chromosomes that are going to go into a second daughter cell. So each of these daughter cells is going to have the exact same hand of cards, so to speak. Okay? So now we've got another cell that is back into G1 phase. This is just where we started. Okay, and if we wanted this cell to go through meiosis, meiosis is a different type of cell division. Meiosis, what it does is it takes diploid cells and makes them into haploid daughter cells. So typically we start off with one diploid cell and we end up with 4 haploid daughter cells. Although your results may vary for reasons I probably won't get into in this particular little card model that I'm doing here. Okay, so in order to go through meiosis, we have to go back to replicating the DNA, and we've seen how I did that before just by throwing these extra cards in there. So we'll scramble them up. So we've gone through S of interphase again, but now for meiosis, we're going to do things a little bit differently than we did in mitosis. So now we're going to go through meiosis 1, because there's 2 parts of meiosis. I'm going to deal out my cards so that everybody is duplicated and together. But this time, this is how we set up for mitosis. For meiosis, watch how it's different. So for meiosis, the homologs are going to line up together. So now you can see I've got 4 aces, 2 copies of the ace of diamonds, 2 copies of the ace of clubs. They're going to go together in one row here. The 2s, 2 of diamonds, 2 of clubs, they're going to go together in a second row, and the 3s are going to do the same thing. So now you can see that I've got 3 sets of 4 cards representing homologous chromosomes. So the aces are homologs, the 2s are homologs, the 3s are homologs, and each of these sets of tetrads is going to have 1 copy of a chromosome from dad, which are the black cards, and 1 set of the chromosomes from mom, which are the red cards. Okay, another thing we should notice here before we go to anaphase these things, besides having these 3 sets of tetrads instead of 6 sets of sister chromatids, is the way that I've lined them up. And you can see that for this, for the aces, I put the club up and the diamond down. Club up, diamond down. For the 2 position, I said, okay, well this time let's put 2 up and club down. And for the 3s, I did the same thing. Diamond up, club down. That's because this is what actually happens in meiosis, is that we don't always get it to be where our chromosomes have obsessive compulsive disorder and they have to line up like this. There's a chance that they could line up like that, but there's only a 1 in 2 to the 3rd, because there's 3, n equals 3, 2 to the n, different ways that we could randomly, independently assort these cards. So what's happening at chromosome position ace has no effect on what's happening on position 2 or position 3. So what's 2 to the 3rd? 2 times 2 is 4, times 2 is 8. So there's 8 different ways that I could break these cards apart at anaphase. Another thing that happens in meiosis, that I can't model here is crossing over. So maybe like a corner of this ace gets swapped with a corner of this ace. So now I have a brand new card that's like 3/4 diamond and 1/4 club. And I have a reciprocal card that's 3/4 club and 1/4 diamond. That process is called synapsis and crossing over. And that would involve me cutting up the cards and taping them back together, which, I might do, but I'm not doing that right now. So we're just gonna assume that that's not happened here, even though under normal circumstances it does, and it increases the amount of genetic diversity that we get. Okay, so this is meiosis 1, metaphase plate. Metaphase plate is running along here. And we're going to anaphase these cards. So bloop and blop. Okay. So you'll see in one of our daughter cells, it looks like we're back to having 6 chromosomes. But we don't have 6 chromosomes. We have 3 chromosomes that have copies, pairs of sister chromatids in them. We have cut the number of chromosomes in half. And this is typically a very confusing point for students who are trying to learn about how we change in ploidy between meiosis 1 and meiosis 2. But it's very important that you know that this reduction division, or this cutting of the number of chromosomes in half, that occurs in meiosis 1. So now this daughter cell, it doesn't have 2 copies of the ACE of the ACE. It's only got one version of the ACE. So if dad's ACE chromosome had a gene for Huntington's disease and mom's didn't, well, this daughter cell has gotten rid of that other allele, that other version of that gene. And if this 2 chromosome had the gene for, let's say, widow's peak, and mom had widow's peak and dad didn't, well, now this cell is only going to have widow's peak and not have that potential to not have widow's peak. And if on chromosome 3 we had a gene for something else, like polydactyly, having extra fingers, and let's say dad had that and mom didn't, and now this cell has only the version of that gene which is no extra digits, no polydactyly. Okay, so we've cut the number of chromosomes in half. All right, so that is that's meiosis 1, and we could do meiosis 2 showing both of the daughter cells that are going to give us 4, but instead I'm just going to show you what's going to happen in one of these. So we don't replicate the DNA during interkinesis in meiosis. What does happen is at metaphase 2 of meiosis, the sister chromatids line up on the metaphase plate, kind of like they did in mitosis, except instead of having 2 copies of the ACE, 2 copies of the 2, 2 copies of the 3, instead of having 2n sets of sister chromatids lined up, now we only have n sets of sister chromatids lined up. We're down to half that number, a single set, the haploid number, so 3. So at anaphase, we're going to have that happening. So our daughter cell that results from meiosis 2, we're only going to end up with 3 cards, no duplicates, no duplicated anything. We've only got 1 ACE, 1 2, 1 3. That's it. So we've got a haploid cell with n chromosomes that are unreplicated. And in order to produce the next generation, we would have to fertilize that haploid cell with another haploid cell that had 1 copy of unreplicated chromosomes. And so maybe we get this. Maybe we get ACE2-3, but we've got the same genes here as on there. So we end up with a different type of daughter cell than we started off with. So all the loci that are heterozygous on the ACE, they're still heterozygous. All the ones that were heterozygous on the 2, now they're all homozygous because we have 2 exact copies of the 2 of clubs, and heterozygous for chromosome 3. Okay, so what this is meant to show you is, where do we divide that ploidy in half? Is when we reduce the number of chromosomes in half, so only have 1 copy of each. It's supposed to show how the metaphase plate is different between mitosis, diploid mitosis, meiosis 1 and meiosis 2. Meiosis is always going to start with the diploid cell and always going to end with haploid cells. That's its function. It's going to give you a different set of genes compared to the parents, because it has to. And that happens for a number of reasons. One is just cutting the number of chromosomes in half means half of your genes are effectively getting left behind in a sense. But it also means that we're making new combinations of chromosomes. So that's another thing that's going to happen. And meiosis 2 is always going to take the haploid number of chromosomes and divide sister chromatids in half, resulting in 4 haploid cells, each of which has n chromosomes that are unreplicated. So one thing I didn't talk about, I'm just going to make a brief comment here, is that you can have mitosis in haploid cells. You can have mitosis in haploid cells. What would that look like? Well, we're going to take our haploid cell, we're going to have to copy our chromosomes, right, during S phase, and it's going to end up looking just like metaphase of meiosis 2, because the sister chromatids are going to separate, and each daughter cell is going to get 3 chromosomes and end up having the exact same complement of chromosomes unreplicated. Now, that doesn't really happen in animals very often, but it does happen in fungi a lot. It happens in plants a lot. So it can happen. So you should know that haploid mitosis and diploid mitosis are going to look different. But for the most part, with me anyway, if I'm going to ask you to show what phase of metaphase we're looking at, it's going to be diploid mitosis and normal meiosis, where in meiosis 1 we go from diploid to haploid, and meiosis 2 we go from haploid to having daughter cells with 1 copy of each chromosome. And I know I said this wasn't a magic trick, but okay, fine.